Hello everyone, welcome to my channel Anupma Biology Classes. In this video, our topic is photoperiodism and vernalization. This video is taken from Plant Physiology Series CBSE Class 11 Topics Part 6 of Chapter 16. Now, let's see here the lecture overview. So in this video, we will focus on photoperiodism and flowering, classification of photoperiodic responses, differences between short-day plant and long-day plant, characteristics of the photoperiodic responses, and vernalization. Now, let's start the topic. Here, our first topic is photoperiodism and flowering. The concept of photoperiodism was given by W. W. Garner and H. Allard of U.S. Department of Agriculture studied flowering in Maryland mammoth variety of tobacco plant in 1920. Variety was a single gene mutant tobacco that did not flower in the spring or summer like wild type. Under controlled experiment in tight boxes where they could manipulate the amount of light and dark, they discovered that flowering only occurred if the day length was 14 hours or less. They called the Maryland mammoth a short day plant because it requires a light period shorter than a critical length to flower. So flowering is an important phase in the life cycle of a plant, transition from vegetative phase to reproductive phases. Physiological chemical mechanism responsible for flowering is controlled by photoperiodism and vernalization. Length of day period has marked effect on the flowering. Garner and Allard in 1920 discovered photoperiodism. And the definition of photoperiodism is response of light to relative length of day and night. Mechanism of photoperiodism is this process is involved following steps like general principle perception, transmission, evocation, etc. In evocation, there are two important aspects in photoperiodism, that is phytochrome, which is a photo or light receptor, and the second is the stimulus. So, because of some scientists which refer to the photoperiod that promotes flowering, and because of this they found a number of other plants which initiates flowering under short day. However, several others remains vegetative under short day but flowered when exposed to long day. So Garner and Allard called such plants as long day plants. So this is all about the discovery and history of the photoperiodism. Now the classification of photoperiodic responses. So Garner and Allard in 1920 recognized three classes of plants according to their photoperiodic responses, and these are short day plants, long day plant, and day neutral plant. Now let's see here the first one, that is the short day plants. A short day plant is one that flowers on photoperiod shorter than the critical day length. For example, Maryland mammoth variety of tobacco flowers when it gets photoperiodic induction of less than 12 hours light and more than 12 hours dark in 24 hours cycle. If the same plant gets photoperiod of more than 12 hours light and less than 12 hours dark, it fails to flower. Various experiments performed using short day plants Xanthium indicates that it is the long and uninterrupted dark period which is important in these plants. This was demonstrated by interrupting the light and dark periods. Interruption of light period by the dark had little or no effect. If the plant get at least 2 hours of light intensity, uh, of light immediately preceding the long day dark period, flowering is unaffected. However, if the long dark period is interrupted by even a brief flash of light, the flowering is inhibited. Long and uninterrupted dark periods are important so much so that some of the short day plants can be induced to flower under continuous dark by supplying them sucrose in the media. Owing to the importance of the long nights, it will be appropriate to call a short day plant a long night plant. Some examples of strict short-day plants are Nicotiana tabacum, which you can see in this picture, then Xanthium istramarium, then Chenopodium, then Glycine max, then Cosmos, Chrysanthemum, and sugarcane, etc. Now, after this, long day plant. A long day plant is one that flowers on photoperiods longer than critical day length. Long days of summer provide the long day plant's perfect photoperiod to permit flowering. These plants require a relatively small dark period. In fact, some of the plants flower best under continuous light. The long dark periods has inhibitory effect on flowering in the long day plants if it is followed by a short light period, but interruption of the long dark period by a brief flash of light initiates flowering in these plants. The long day plants can flower under short day conditions if the short light period is followed by still shorter dark period. For example, several long day plants flower under periodic cycles of 7 hours of light alternating with the 5 hours of darkness, or even 4 hours of light alternating with 2 hours of darkness. As a case of the short day plant, the flowering in long day plants is not determined by the period of day, but it is determined by the period of short night. Thus, it will be appropriate to call a long day plant a short night plant. Some common examples of the strict long day plants are Hyos Cyamus niger, that is henbane, Avena sativa, that is oat, Dianthus superbus, that is carnation, radish, spinach, etc. After this, the third one, that is the day neutral plant. These plants do not require specific photoperiods to flower. Such plants flower in almost all the photoperiods ranging from a few hours to 24 hours of uninterrupted light periods. The day neutral plants are also known as photoneutral or intermediate plants. So here you can see that in case of day neutral plants, day length immaterial than 12 hours for the flowering. So the common examples like the cucumis cucumber, gia maize, balsam, tomato, cotton, sunflower. These are the examples of the day neutral plants. Now, let's see here some key biological process, that is the photoperiodism. So first one, that is the long day plants, short day plants. So, early summer, short periods of darkness induce flowering in the long day plants such as iris, but not in the short day plants such as goldenrod. Late fall, long periods of darkness induce flowering in the short day plants such as goldenrod, but not in the long day plants such as iris. Interrupted night. If the long night of the winter is artificially interrupted by a flash of light, the goldenrod will not bloom and then iris will. So here you can see the long day, that means short night plant, and the short day, that means long night plant, and the critical night length. So here you can see the day length requirement for the flowering in the three categories of the plants. Now, types of flowering in angiosperm. Long day plants where the flowers when day are the longest and nights are shortest, that is the midsummer, and the examples are radish, spinach and lettuce. Short day plants where the flowering in spring, late summer and autumn when days are shorter, like the poinsettias, chrysanthemum and asters, and the day neutral plants like the flower without regard to the day length, like the roses, dandelions and tomatoes. Now, what are the differences between the short day and the long day plants? Short day plants where the plant flower when photoperiod is less than the critical day length, while the long day plants where the plant flowers when the photoperiod is more than the critical day length. In short day plant, interruption during light period with darkness does not inhibit flowering, while in long day plants, interruption during light period with darkness inhibit flowering. In the short day plant, flowering is inhibited if the long dark period is interrupted by a flash of light, while in the long day plant, flowering occurs if the long dark period is interrupted by a flash of light. In short day plant, long continuous and uninterrupted dark period is critical for flowering, while in the long day plant, dark period is not critical for the flowering. In short day plants, flowering does not occur under alternating cycles of the short day and short light period, while in the long day plants, plants flower when the photoperiod is flowering occur under alternating cycles of the short day followed by the still shorter dark period. So these are the main differences between the short day plants and the long day plants. Now, characteristics of the photoperiodic responses. So, the first one, that is the genetic control. It is now well known that the photoperiodic responses are under the control of genes. This can be modified by various genetic manipulations to yield such varieties of plant which can flower under desired photoperiods. For instance, investigators at the National Botanical Research Institute, Lucknow, have been able to evolve varieties of chrysanthemum by plant breeding experiments which can flower in the different months of the year, including summer. Further investigations are in the progress to modulate flowering in some economically important crop plants. Second is critical day length. It is the duration of light period required to induce flowering which must not be exceeded in case of short day plants and should always be exceeded in case of long day plants. For example, Xanthemum, a short day plant, require a critical day length of 15.5 hours. If these plants get less than 8.5 hours dark, it fails to flower. That means the critical day length of 5.5 hours must not be exceeded. On the other hand, Hyoscyamus niger, a long day plant, requires critical day length of 11 hours. The plant still flowers if it gets more than 11 hours photoperiod. That means it is a long day plant. Looking to the above examples, it is clear that there is no relation with the total day length. A plant is called short day if it gets long and uninterrupted critical dark period to flower. Similarly, a plant is called long day if it gets more than the critical day length to flower. Next is inductive cycle. Plants usually remain vegetative if they do not get required photoperiods. For example, Xanthemum, a short day plant, remains vegetative when gets 16 hours light and 8 hours dark. The same plant when gets in single cycle of 15.5 hours light followed by 18.5 hours uninterrupted dark and again interfered to long day conditions, flowering is initiated within the 2 to 3 days. This single cycle of the 15.5 hours light and 8.5 hours dark is an inductive cycle for the Xanthemum. Many plants require more than one inductive cycle to flower. The number of inductive cycles required for flowering varies from 1 to 25. Next is perception of photoperiodic stimulus. It has been experimentally demonstrated that light stimulus is perceived by leaves. Moreover, it has been shown that the photoperiodic stimulus can be transmitted from one branch of the plant to the another or from one plant to the another through graft union. These conclusions are based on the following observations, like a plant from which all the leaves have been removed fails to flower even after getting the required inductive cycles. In Xanthemum, a short day plant, flowering occurred if 1/8 of a leaf was exposed to short days. A single leaf of the plant exposed to required inductive cycles was able to induce flowering when it was grafted onto a plant kept under non-inductive conditions. The floral stimulus moves from one species to another through a graft union if an inductive twig of Xanthemum was grafted onto a vegetative soybean plant. So, this is all about the perception of the photoperiodic stimulus. Now, role of phytochrome in the flower initiation. Following observations, here, indicate that phytochromes is involved in the initiation of the flowering stimulus in plants. And these observations are: long dark periods of the short day plants, if interrupted midway by a brief exposure of red light, the flowering is inhibited. If the exposure of red light is immediately followed by far red light, the flowering is promoted. Whether the flowering is promoted or inhibited depends on the exposure given in the last when the two types of radiations given successively. So visible radiations before it can act must be observed by some pigment present in the plant. This above observation indicates the presence of the some pigments in the leaf which must be photoreversible. Several experiments indicate that light is absorbed by a photoreversible pigment phytochrome and the flowering is a phytochrome mediated process. And the last here, which is a characteristic of photoperiodic responses, the flowering stimulus, that is floragen. It has been suggested by several authors that some flowers promoting stimulus is formed in the leaves and then conveyed to meristems. Chalikian gave the view that the flower hormone, namely floragen, is synthesized in the leaves under favorable photoperiodic conditions. This hormone is transmitted to the growing point where the flowering occurs. Unfortunately, floragen has never been isolated. Now, vernalization. Many species, especially many biennials and perennials, are induced or promoted to flower by the exposure to low temperature. They grow vegetatively during the warm season, receive the low temperature treatment during the winter, and grow further to bear flowers and fruits in the following warm seasons. Lysenko, a Russian worker, used the term vernalization for a low temperature promotion of the flowering in case of plants. The low temperature requirement for the flowering was first noticed by Klippart in 1857 while working with two varieties of wheat, the winter wheat and the spring wheat. The winter wheat is sown in winter and produces flowers in summers. The spring wheat is sown in the spring and produces flowers in summer. He observed that if the germinated seeds of the winter wheat are treated with low temperature, they can behave as spring wheat. He concluded that winter wheats require low temperature for subsequent flowering, the phenomena now known as vernalization. In nature, the plants require vernalization commonly behaves as biennials. The biennials complete their life cycle in 2 years. They germinate and grow vegetatively in the first year and produce flowers in the second year of growth. These plants fulfill their cold requirement during winters. If a biennial is maintained in the greenhouse without a cold treatment, it will grow vegetatively for a year. Now let's see the effect of vernalization. So, a vernalized plant, here you can see that there's a slightly germinated set, chilled for a month at 5 degrees Celsius, planted at ordinary temperature, vernalized plant, and no chilling, planted at ordinary temperature, no vernalized plant. The actual site of the cold stimulus in most of the plants is meristematic cells in the bud, apex of embryo, developing leaves, etc. If we talk about the types of vernalization, it is of two types, facultative and obligate. Facultative vernalization, where the flowering will appear earlier once exposed to low temperature, like the winter annual triticale. And the obligate vernalization is must exposure to low temperature to a desired period of time, that is the biennial plants. So vernalization is a requirement of plants to go through a period of cold before they can flower. Vernalization stimulus. Several experiments evidences suggest that the flowering stimulus is synthesized in the meristematic cells as a result of a specific cold treatment. So vernalization stimulus is received by vernaline. Transmission of vernalization stimulus across a graft union in Hyoscyamus niger has been studied by a German botanist, G. Melchers. He took two plants, two branches, one previously vernalized and other unvernalized, and grafted them together, both flowers in appropriate photoperiod conditions, but unvernalized plant alone failed to flower. From above experiment, it is obvious that vernalization stimulus is transmitted from vernalized to unvernalized plant or branch. And the mechanism of vernalization is the stimulus received by the actively dividing cells of the shoot or embryo tip travels to all part of the plants and prepare it to flower. The stimulus has been named as vernaline. It can be passed from one plant to another through grafting in case of henbane but not in others. So, in case of vernalization stimulus, The substance moves from site of perception to the site of flower initiation and results flowering. The German botanist demonstrated that the product of vernalization could be transmitted from vernalized to the unvernalized hyoscyamus plant from a graft union, and he named his substance as vernaline. The vernaline is a hypothetical substance and has not been isolated. So here you can see that experiment to demonstrate that flowering stimulus can translocate from vernalized plant to unvernalized plant through graft union. So now the process of vernalization. First soak the desired seed in water, then allow it to germinate in processing chamber, then subject it to the chilling treatment, then the moistened seeds are dried, then finally the seeds are sown in the fields. So this is the end of video in which we knew about the photoperiodism and vernalization. The next upcoming videos will be related to biological classification. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any question, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.